So permanent tissues are the cells which no longer divide. So why do they don't divide is because the cells are already matured. Whatever food has been synthesized in the plant, it is stored in the parenchyma either in the form of starch or other materials in the parenchyma cells. So tracheids are usually elongated and they are tube-like cells, and they have thick and lignified walls present within them. Hello everyone, a warm welcome to another session on chapter 6, that is Anatomy of Flowering Plants. I am Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysore, Temple of Excellence. So in the previous session, we had studied about some of the important tissue that is involved in a plant. That is especially if the elongation or the growth of the plant has to take place. So we had studied about the meristematic tissue, and in meristematic tissue we had studied about the apical meristem, wherein we studied the root apical meristem and the shoot apical meristem and also the intercalary meristem, and also we learnt about the secondary meristem. So in today's session, we shall learn about some of the other important tissues wherein I had mentioned in the previous session about permanent tissues. So we shall study about that. So the permanent tissues are again divided into simple tissues and complex tissues. So before moving into that, what is permanent tissues? So permanent tissues are the cells which no longer divide. So why do they don't divide is because the cells are already matured. So they have reached a stage wherein the mature cells will no longer divide. So such cells which do not undergo any division are called as the permanent tissues. And in the permanent tissue, there are again two types. One is the simple tissue and the other one is the complex tissue. So simple tissues are the one that are made up of parenchyma, collenchyma and sclerenchyma. So moving on to the complex tissues, it is the xylem and the phloem. So in the xylem and the phloem, again the xylem are made up of xylem as well as the phloem are made up of different components, such as the components of xylem are tracheids, the vessels, the xylem fibers and the xylem parenchyma. And next, moving on to phloem, the phloem is made up of different components which are the sieve tube elements, the companion cells, that is the phloem parenchyma and the phloem fibers. So we shall study each of these one by one in today's session. So first, starting with the simple tissues. So simple tissues means there is no complexity. So when will the complexity occurs, or when will a tissue become complex? If it is made up of two or more different types of cells. But in the case of simple tissues, they are made up of only one type of cells. Say for example, if I say parenchyma, all the parenchymatous cells will look similar. If I say collenchyma, all the collenchymatous cells will look similar. That is, they are isodiametric. So such cells are called as simple tissue. That is, they are made up of only one type of cells. So what are the different types of simple tissues? They are parenchyma, collenchyma and sclerenchyma. So we shall study about them one by one. So first, starting with parenchyma. So parenchyma is one of the major component of the plant cell. So here almost all the organs in a plant body, they are made up of the parenchyma. So you take a section or a cross section of be it a stem, a root or a leaf. The first tissue that you find in a plant, that is in the cortex, is nothing but the parenchyma. And these parenchyma, they are isodiametric cells. That is, they have a similar diameter. Or when you look at the parenchyma cells, their edges or have the similar diameter. They are similar to each other. Each of the cells look similar to each other. Therefore, they are isodiametric in nature. And when we talk about the shape, it is spherical. Sometimes they can either be just spherical, sometimes they can be oval, and sometimes they can be round, they can be polygonal, or they can be elongated in shape. So elongated in shape, for example, I can talk about the palisade parenchyma. So palisade parenchyma are usually elongated in shape. When I talk about spongy parenchyma, they are irregular in shape. So either they can be polygonal, having many angles as in the case of spongy parenchyma, or they can be elongated as in the case of the palisade parenchyma. So this is the shape of the parenchyma cells. And when we talk about the covering or the walls of the parenchyma, they are very thin and they are made up of a major component called cellulose. So we all know that plant contains a lot of cellulose in it, right? That is why animals have the ability to digest the raw plant as it is, because in the digestive system, they have the enzymes that help in the digestion of the cellulose. But we human beings, we cannot eat plant raw. Why? Because we do not have the digestive enzyme that is capable of digesting the cellulose. So therefore, it is because the plants, the cells of the plants are covered by cell walls which are made up of cellulose. So cellulose is one of the major component there. And next, these parenchyma cells, they are very closely packed, or they may have small intercellular spaces. So when I say intercellular spaces, you can see there they are in some. When I talk about collenchyma, they are very closely packed. But in the case of parenchyma, they are closely packed, but also in some cases they may be packed in such a way that they have intercellular spaces. So you can see gaps here. These are nothing but the intercellular. So intercellular spaces are present, and also the cells are isodiametric. That is, some are oval, some are having different angles, some are spherical, some are round, but still the cells look almost similar to each other. Therefore, they are isodiametric in nature. So next, moving on to the function. So the main function of parenchyma is photosynthesis, and especially in parenchyma there are a set of cells called as the Sclerenchyma. So these Sclerenchyma, as the name itself suggests, Sclerenchyma, as the name itself suggests, they have chlorophyll pigment in them, therefore helping in the process of photosynthesis. Plants are able to synthesize their own food, and they also act as a storage structure. So whatever food has been synthesized in the plant, it is stored in the parenchyma, either in the form of starch or other materials in the parenchyma cells, and also they help in the process of secretion of various components in the plant. So this is the function of the simple tissue, that is parenchyma. So next, moving on to the other simple tissue, that is collenchyma. So collenchyma is situated just below the parenchyma. So if you look at a plant cell under the microscope, you can find in the cortex the first layer will be the parenchyma, and right below the parenchyma you will find the collenchyma. So collenchyma, it is a layer below the epidermis in the dicotyledonous plant. So in the epidermis, in the dicotyledonous plants, these collenchyma usually occurs right below the epidermis. But in the case of some other plants, depending on the species, the collenchyma may occur after the parenchyma. And it is found either as a homogeneous layer or in patches. That is, homogeneous layer, one stretch of plant cells may be collenchyma at a stretch, that is homogeneous means the same set of collenchyma cells will be present, or in some cases they may occur just patches here and there in between the parenchyma cells. So therefore, they are found either as a homogeneous layer or they are found either in patches inside a plant cell. And these collenchyma, they consist of cells which are much thickened at the corners, and because they are thickened at the corners, there is no gap between the collenchyma, unlike the parenchyma. So as I showed you, in parenchyma there were intercellular spaces present in between, though their cell was covered with a compound, that is cellulose. But in the case of collenchyma, the cells are thickened in the corner because of excess deposition of the compound cellulose, hemicellulose and pectin, because these three compounds constitute the cell wall, therefore resulting in thickening of the cell wall. That is why in between the cells there is no intercellular spaces. Therefore, collenchyma is a cell that is tightly packed. They are tightly packed cells. They occur in groups wherein they are tightly packed. So you can see here, collenchyma, they have a protoplasm. So what is a protoplasm? It is nothing but a colorless substance that is present within the cytoplasm. So they have a protoplasm, then they have the cell wall. So what is the cell wall made up of? It is made up of pectin, cellulose and hemicellulose. So therefore, because three of the deposition is present, that is deposition of pectin, cellulose and hemicellulose, therefore because it will result in thickening of the cell wall, and because of the thickening of the cell wall, there is no space between the collenchyma cells. Therefore they have a very thick corner. So they have thick corners. That is why there is no intercellular spaces. So this is about collenchyma. So next, moving on to the shape of the collenchyma. So collenchyma can either be oval, spherical or polygonal, that is having many angles, and often they contain chloroplast. So because of the presence of the chloroplast in the collenchyma, that is why they help in the process of the photosynthesis, and these cells also assimilate food, that is they help in the manufacturing of food because they have the chlorophyll pigment in them, that is, and also because they have the chloroplasts are the ones which contain the chlorophyll pigment, which are responsible for the assimilation of the food or the synthesis of the food by the plants. And also, as I told you, intercellular spaces are absent. Why are intercellular spaces absent? Because there is cell wall thickenings present, wherein the cell walls will have depositions of cellulose, hemicellulose and pectin substances. And moving on to the function, these collenchyma cells, they actually provide mechanical support or rigidity to the growing parts of the plant, such as the young stem and the petiole of the leaf. So the young stem and the petiole, they are usually fragile. So at such cases, these cells, that is the collenchyma, they provide mechanical support. So you know that the stem has to stand upright. So if it has to stand upright, it needs to be strong and hard. So that function is actually supported by the collenchyma. So this is about the shape. So you can see it is either oval or it may be spherical, or also they may have polygonal shapes having many angles, and they have chloroplast within them. So because of the presence of the chloroplast, they are usually green in color, and therefore they help in the process of the photosynthesis. So next, moving on to the next simple tissue, that is sclerenchyma. So sclera means dead. So sclerenchyma are the cells which are dead. So you can imagine, if they are dead, what will they have a function? No, these tissues, even in spite they are dead, they are very rigid and they provide mechanical support to the plant. So whatever excess mechanical support is needed to the plant for the proper structure of the plant and all that, that is provided by the sclerenchyma. So the sclerenchyma, it is made up of cells which are long and narrow. So you can see here, they are very long and they are very narrow. They are not broad as like that of the parenchyma or the sclerenchyma, but they are narrow and they are very long. And they have thick and lignified cell wall. So lignified cell wall means the cell wall is deposited with lignin. Like how in collenchyma I told you the cell walls are deposited with the pectin, hemicellulose and cellulose. Likewise in the sclerenchyma, the cell walls of the sclerenchyma, which are narrow and long, they are made up of lignin. They are very thick. Why are they thick? Because of the lignin deposition. And the cell walls, they have either few or numerous pits or openings present in them, or pores present in them, and they are usually dead without any protoplast. So what are protoplasts which do not have a cell wall are called as the protoplasts. So they do not have a protoplast, and also these sclerenchyma are dead tissues. Though they are dead tissues, they provide excess mechanical support to the plants. So on the basis of different forms of structures or origin and development, the sclerenchyma are usually of two types. So depending on the structure, how the sclerenchyma looks and how they are formed, from what kind of cells they are formed, based on all those, they are of two types, that is they are fibers or in the form of sclerites. So first we shall see how Sclerenchyma fibers look like. So they are usually again thick-walled. So the Sclerenchyma fibers are also thick-walled. They are elongated, they are narrow, and they are tapered at each ends. That means they have a pointed end. So they are pointed cells, and they usually occur in groups in various parts of the plant. So fibers usually provide rigidity to the plant. We all know that. And here we have taken the example of a fiber. So they usually the fiber, Sclerenchyma fiber, so it can be either Sclerenchyma fiber or Sclerenchyma sclerites. So here we are talking about Sclerenchyma fiber. So in Sclerenchyma fibers there is a lumen or a cavity that is present, and the cell is entirely, it is made up of a group of cells, and each cell, they are elongated, they are narrow, they have tapering ends which are pointed. So they have pointed ends, and the thick cell wall is very, very thick. So this is about the Sclerenchyma fiber. So next, moving on to the sclerites, which are another form of the Sclerenchyma apart from the fibers. So sclerites, they are usually spherical, oval or cylindrical. So what did I tell you? The type of Sclerenchyma cells are based on the shape of the cell. So earlier the fibers were narrow, they were elongated, having pointed ends. But when I talk about sclerites, it is a completely different shape, opposite shape of the fiber. So they are spherical, they are oval, and they are sometimes they can be cylindrical as well. And they have thickened dead cells with very narrow cavity. So earlier fibers also had a narrow cavity, and similarly the sclerites also have a narrow cavity which contains dead cells. Therefore, because Sclerenchyma fibers has lumen filled with dead cells and sclerites also have lumen filled with dead cells, therefore Sclerenchyma is a dead tissue. That is why it is a dead tissue. So they have narrow cavities which are filled with thickened dead cells, and they are commonly found in the fruit walls of nuts. So these sclerites are usually found in the walls of the fruits, these nutty fruits. They are found in the fruit walls of nuts, in the pulps of fruit like guava, pear, and in sapota, and they are also found in the seed coats of the leguminous plant, and also they are found in the leaves of the tea plant. So these are the common plants in which parts of the plants in which the Sclerenchyma are produced. So the main function of especially the sclerites Sclerenchyma are found, and the function is they provide mechanical support to the organ. So for the movement of the plant organ or whatever, and for the rigidity of the plant organ, or for the plant organ to stand upright, for all that these Sclerenchyma usually helps. So next, moving on to the complex tissues. So we finished with the simple tissues, which was the parenchyma, the collenchyma, and the Sclerenchyma. So now moving on to the complex tissues. So complex tissues are the xylem and the phloem. So what is the xylem and phloem? So the vascular tissues in a plant body is nothing but the xylem and the phloem. So the xylem helps in the conduction of the water, and the phloem helps in the conduction of the food. So xylem, it is again composed of 4 different elements, or they are made up of 4 different components or xylem elements, which are called as the tracheids, the vessels, that is the xylem tracheids, the xylem vessels, the xylem fibers, and the xylem parenchyma. And usually in the case of gymnosperms, so all the other plant species have tracheids, vessels, xylem fibers, and xylem parenchyma in the xylem cell, but the gymnosperms, they lack the vessels in their xylem. So gymnosperms only have tracheids, xylem fibers, and xylem parenchyma. In gymnosperms, the vessels are absent, but all the other plants have the rest of the xylem components or the xylem elements along with that of the vessels. So this is one peculiar feature of the gymnosperm members. So gymnosperms are nothing but those plants which produce cones. They are the non-flowering plants which produce cones as their reproductive structure, and also they have naked seeds, that is the seeds which are not covered by any of the fruit wall or anything. Those are gymnosperms. And next, moving on to the function of xylem. So xylem usually helps in the transport of water from the roots towards different parts of the body. Therefore, throughout the plant body, in the cells of the plant body, we can find the xylem tissue. They help in conducting. So that is why they are called as the conducting tissue for water. That is, they conduct or they carry water from the roots to different parts of the plant body. So they are the conducting tissue for water, and also they help in the carrying of minerals along with the water from the roots to the stem and the leaves of different parts or to different parts of the plant body. Next, they also provide mechanical strength to the plant part. So xylem also provides mechanical strength. Why? Because xylem have xylem fibers in them. So these xylem fibers are the ones which usually provide the mechanical strength to the plant body. So these are the functions of the xylem. So next we shall study the components or the elements of xylem one by one. So I told you what are the elements of xylem. They are tracheids, vessels, xylem fibers and xylem parenchyma. I had told you one more thing, that in gymnosperms vessels are absent, but all the other three xylem elements are present. So tracheids. So tracheids are usually elongated and they are tube-like cells, and they have thick and lignified walls present within them. So remember, we had talked about Sclerenchyma fibers. So how the Sclerenchyma fibers look like, just like that in a simple tissue, just like the xylem tracheids also looks like. So they are elongated or they are tube-like. So as you can see, they are elongated and they are tube-like. So, and their wall is made up of lignin. They have lignified walls. That is, the covering is made up of lignin content. Their wall is made up of lignin. That is why it is called as they have lignified walls. So, and thick and lignified walls, and they have tapering ends. So tapering end means, for example, here the ends of the tracheids, they actually join to form a pointed end. So they have a tapering end, and these are also dead and without protoplasm. That is, protoplasm is nothing but a colorless fluid that is present within the cytoplasm of a cell and having all the organelles. So these are dead tissues because they lack a protoplasm. That is, they do not have a protoplasm and they are the dead tissues. And the inner layers of the cell walls, they have thickenings which vary in form. So apart from having an outer layer, it also has an inner layer. So the outer layer is made up of lignin. That is why it is called as lignified walls. And the inner layer is also have some thickenings. That is, these coverings, they actually vary in shape and size or vary in form. And in flowering plants, the tracheids and the vessels, they are the main water transporting elements. So in xylem, the components of the xylem or the elements of the xylem, that is the tracheids and the vessels, are the ones which actually help in the transport of water from one part of the body to all the other different parts of the plant body. This is one example of a tracheid. So tracheids also, you can see pits in the tracheid. So why pits might occur is because of the unequal thickenings, that is the inner layers which have thickenings in the tracheids. That is why. So next, moving on to the xylem vessels. So vessels is also one of the component of the xylem or the element of xylem. So these vessels also, just like the tracheids, they are long and cylindrical tube-like structure, and they are made up of many number of cells called as vessels. So they have many cells called as vessels, and also they are lignified. That is, the wall is made up of a lignin, and they have a central cavity. So this vessel, they have a central cavity, that is a lumen or a central cavity which is covered by lignified walls. And just like the tracheids, they are also long cylindrical tube-like structures, and these also, they do not have a protoplasm. They are devoid of the colorless liquid that is present in the cytoplasm, which is nothing but the protoplasm. And they are interconnected through perforations in the common wall. So how the exchange of the components through different elements takes place is because each xylem vessel is interconnected through the perforations or the openings that are present in their wall. So the presence of vessel is a characteristic feature of the angiosperms. Why? Because in gymnosperms, the vessels are absent. So therefore, the presence of vessels in a plant body or in the cells of the plant, especially in the xylem, means that that particular plant belongs to a angiosperm, that is a flowering plant, and that is not a gymnosperm. So therefore, the presence of vessels is a characteristic feature of the angiosperm. So this was about the vessel. So next, moving on to two important components or elements of the xylem tissue, that is the xylem fibers and the xylem parenchyma. So the xylem fibers, they have thick walls and they have central lumen present in them, that is a cavity present in them. So as you can see, they have thick walls and they have cavity or central lumen present, and these may either be septate or they may be aseptate. So here I have shown you the xylem fiber, which is aseptate, and they are long, they are lengthy, cylindrical, having a lumen cavity and also tapering at the ends or pointed at the ends. And they can either be septate, that is having septars, or they can also be aseptate, that is without any septars. Next, moving on to xylem parenchyma. So these xylem parenchyma, they are living. So as I told you, the tracheids and the vessels, they were dead tissues. Unlike those two, these xylem parenchyma, they are living and they have very thin wall. So far, the xylem fiber, the tracheids and the vessel had thick lignified walls, right? But in the case of xylem parenchyma, the cells are living and they have very thin wall and their walls are made up of cellulose. So cellulose depositions are present in parenchyma. So anywhere the parenchyma cells, they have a cellulose deposition. So first, when talking about the simple tissue, I had told you about parenchyma, wherein I had mentioned that the parenchyma have a thin wall and the wall is covered by cellulose. Similarly here, xylem parenchyma also have cellulose in their wall. So these xylem parenchyma, they are somewhat elongated and they have a cytoplasm. They have a centrally located nucleus. Nucleus is present. Cytoplasm is also present in them. They store food materials in the form of starch or fat, and also they store other substances such as tannin. Tannin is one of the important component, or it is a secondary metabolite that is produced by the plant body, and these tannins actually protect the plant against the herbivorous animals or other insects. So tannins also play a very important role in the defense strategy in the plant. So these are the components that are stored. And I remember I had told you xylem parenchyma, the main function of parenchyma in general is to store food. So in the case of xylem parenchyma, which is a component of the xylem tissue, there these parenchymas, they help in the storage of the food materials such as starch, or in the form of fat, or in the form of food materials such as tannins also are stored in it. So and also they help in the conduction of water that takes place by the ray parenchymatous cells. So whatever water, it is also one of the water conducting cells. So in xylem, the tracheids as well as the xylem parenchyma, they help in the conduction of the water. So these are some of the important characteristics of the different elements that are present in the xylem, which is a water conducting tissue. So next, moving on to the food vessels. So vessels, they are usually long, cylindrical and tube-like structures, and they are made up of many cells which are called as vessel members. So each of these vessel members, they come together to form a vessel, and they are lignified. They have lignified walls and they have a central cavity, like how the tracheids, all that had the lignification on their walls or lignin deposition in the wall, unlike the parenchyma wherein there was cellulose deposition. Just like that, these vessels also have lignin depositions on their wall, and they also have a lumen, that is they have a large central cavity being present. So you can see they have a large central cavity, and they do not have a protoplasm. They are devoid of protoplasm, and the vessel members are interconnected through perforations in their common wall. So, perforations or small openings are being present through which they interconnected, or the exchange of material take place through these perforations, because xylem vessels, I told you, they are usually present in groups, and the presence of vessels is actually one of the characteristic feature of the angiosperms. So next, moving on to xylem. Again in xylem, it is made up of primary xylem. So what are these primary xylem? So primary xylems are the first formed xylems. Again in the primary xylem, there is this protoxylem and the metaxylem. So protoxylem are the first formed primary xylems, and the metaxylem is the one that is formed after the formation of the protoxylem. So protoxylem are first formed primary xylem elements, and metaxylem are the later formed primary xylem elements. So you can see here very clearly the protoxylem will be small. Why? Because they are still in a developing stage, because they are the first form. These protoxylem later on mature to form the metaxylem. So the protoxylem are first formed, and after the formation of the protoxylem, the metaxylem gets formed. So these are it is nothing but the size of the xylem and also the position of the xylem determines whether it is a protoxylem or a metaxylem. So in metaxylem, it is usually in stems, the protoxylem lies towards the center. So when we take a cross section of the stem, say for example, I have taken a cross section of the stem. So stem usually looks cylindrical. So if this is the pith region, of the stem, the protoxylem actually lies towards the pith. So protoxylem will be towards the pith and metaxylem will be towards the periphery. So this will be the metaxylem and this will be the protoxylem. So in stems, the protoxylem lies towards the center, that is the pith, and the metaxylem lies towards the periphery of the organ. So this type of primary xylem. So now consider, I've taken the cross section of a stem. So while looking, this is the pith region. So if the protoxylem is found present towards the pith and the metaxylem is present towards the periphery, then such type of arrangement or primary xylem is called as endark. Endark means, endark is a condition in which the protoxylem is situated towards the pith and the metaxylem is situated towards the periphery or towards the sides of the plant organ. That is endark. So metaxylem is again, or the xylem is again of two types depending on the arrangement of the protoxylem or the position of the protoxylem and the metaxylem. They are nothing but endark, and one more is exarch. So endark condition is usually seen in the stem. So say for example, somebody has taken a cross section and kept the material under the microscope. So as soon as you see, if you see that the protoxylem is present towards the pith and the metaxylem is present towards the periphery, then it is understood that that particular plant material which they have kept under the microscope, it is a stem. And if it is a root, it is vice versa. If it is root, say consider again this to be a root. Metaxylem will be present towards the pith and the protoxylem will be present towards the periphery. So this will be the metaxylem and this is the protoxylem. So such type of xylem arrangement, it is called as exarch. So especially we need to consider the position of the protoxylem here. So protoxylem is inside towards the pith. So it is endark. Here protoxylem is outside towards the periphery. Therefore it is exarch. So in roots, usually the exarch condition of xylem arrangement is seen, wherein the protoxylem lies towards the periphery and the metaxylem lies towards the center, that is towards the pith. So such arrangement of the primary xylem is called as the exarch. So what did we learn here? Primary xylem is of two types, that is protoxylem and the metaxylem. So depending on the position of the protoxylem and the metaxylem, we can find out whether protoxylem usually will be present towards the center and the metaxylem will be arranged towards the periphery. Such a primary xylem is called as the endarch. And in the case of the roots, the metaxylem will be present towards the center or towards the pith and the protoxylem will be present towards the periphery. Such a condition is called as exarch. So next, moving on to the one more complex tissue, that is phloem. So what did I tell? Permanent tissues. So what are permanent tissues? Those tissues which have stopped dividing, they will no longer divide. In the permanent tissue, we had the simple permanent tissues and the complex permanent tissue. So in simple permanent tissue, we had finished studying about the parenchyma, the collenchyma and the sclerenchyma. So moving on to the complex permanent tissue, we studied about the xylem. So next we'll study about the phloem, which is also one of the complex permanent tissue. So the phloem usually helps in the transport of food materials, that is from the leaves to other parts of the plant body. So food that is synthesized by the leaves, that will be carried or transported to different parts of the plant body through cells called as the phloem or tissues called as the phloem. So phloem in angiosperms, it is usually made up of, they have certain components or elements called as the sieve tube elements, the companion cells, the phloem parenchyma and the phloem fibers. But in gymnosperms, they have albuminous cells and sieve cells. So along with that, in gymnosperms, the phloem elements are somewhat extra. They have albuminous cells and they have the sieve cells being present, but they lack the sieve tubes and the companion cells. So what did I say? I'm talking about the angiosperms and the gymnosperms. So in angiosperms, the phloem is made up of the different components called as the sieve tubes, the companion cells, and the phloem parenchyma and the phloem fibers. In gymnosperms, the sieve tubes are not there. Instead of the sieve tubes, sieve cells are present and the companion cells are also not there. So instead of a companion cells, albuminous cells are present. They have albumin, that is endospermous cells. Albuminous cells are present. And rest, that is the phloem parenchyma and the phloem fibers are present. So angiosperms have sieve tubes, companion cells, phloem parenchyma and phloem fiber, whereas the gymnosperms, they have the sieve cells, the albuminous cells, the phloem parenchyma and the phloem fiber. So this is peculiar about the gymnosperms. So unlike how gymnosperms, they did not have the vessels, xylem vessels, right? Here, they don't have two components of the phloem, but instead of the two components of the phloem, they have the sieve cells and the albuminous cells instead of the sieve tubes and the companion cells. So next, moving on to studying each of the components of the phloem tissue one by one, that is the sieve tube elements. So the sieve tube elements, they are usually long, they are tube-like structures and they are arranged longitudinally and they are always with the companion cells. So the sieve tubes, they do not exist alone. They are always grouped with the companion cell or they exist only with the companion cells. So they are long. So you can see this is a phloem tissue. So the phloem tissue is made up of sieve tube. It is made up of phloem parenchyma and it is also made up of the companion cells and it is also made up of the phloem fibers. So the sieve tube elements, they are usually long. So you can see the sieve tube elements, they are usually long and they are cylindrical and also they are tube-like structures and they are arranged longitudinally along with the companion cell. So these companion cells and the sieve tubes, they lie close to one another. They are always associated with each other, and the end walls are perforated in a sieve-like manner to form the sieve plates. So you might have seen the sieves with openings, they have pores, right? So filter something, just like that the end walls. So the end walls of the sieve tube elements, they also have numerous perforations or numerous pores which are present, that forms a sieve plate. So they have this sieve tube elements at the end, they have sieve plates or they have perforations being present. So sieve plates, as you can see, they have sieve plates present which has perforation. So therefore they are called as the sieve plates. So just like, imagine something that is used for sieving. So just like that. And a mature sieve element, they usually possess a cytoplasm at the periphery, that is at the corners. They usually have the cytoplasm and they have a large vacuole, but they do not have a nucleus. So these are some of the features of the sieve tube elements. So they have cytoplasm. At the periphery, they have cytoplasm. So unlike the other cells of a plant, like say for example parenchyma, they are not completely filled with cytoplasm, but cytoplasm is present only at the periphery, and also they have a large vacuole. So large vacuoles are present. So large vacuoles or openings are present, but they do not have a nucleus, they lack a nucleus, and the functions of the sieve tube are to control the nucleus of the companion cells. So that is why I told you the sieve tubes and the companion cells are always associated with each other. Why? Because the companion cells, as you can see, companion cells has nucleus. So the main function of this particular sieve tube element is to control the nucleus or control the functioning of the nucleus of the companion cells. So this is the characteristic features of the sieve tube elements. So next, moving on to the type of cells, that is the companion cell. So we talked about the sieve tube elements. So sieve tube element is always associated along with the companion cells. Why? Because the nucleus of the companion cells is always controlled by the sieve tube elements of the phloem. So next, moving on to the companion cells. So companion cells, they are usually parenchymatous cells and they are usually closely associated with the sieve tube elements, and the sieve tube elements and the companion cells, they are usually connected to one another. That is, there is a pit that is present with each other wherein the exchange of the materials and all those takes place, and the companion cells, they help in maintaining the pressure gradient in the sieve tube. So whatever the external concentration of the solution might be, whatever it is, it helps in maintaining the turgor pressure gradient, or it helps in maintaining the pressure gradient in the sieve tube element. So that is one of the functions of the companion. So sieve tube elements, they control the functioning of the nucleus of the companion cells. The companion cells in turn try to maintain a pressure gradient in the sieve tube element. So therefore they are closely associated to one another. So if you see, we have the companion cells here. So they have elongated parenchyma cells. So they have, the cells are parenchymatous. They have parenchyma cells and they have a nucleus, and they are elongated and they have tapering ends. And next, moving on to phloem parenchyma. Phloem parenchyma, they are also elongated, they have tapering ends, and they are usually cylindrical in shape, and they have a dense cytoplasm and a nucleus. So as you can see, the phloem parenchyma, they are elongated, they are tube-like, they have dense cytoplasm, that is very thick cytoplasm is being present, and also they have a nucleus. The cell has a nucleus. So next, moving on to the cell wall deposition. So the cell wall is composed of cellulose, that is, the wall is made up of cellulose or it has cellulose deposition, and they have pit through which the plasmodesmatal connections takes place between the cells. So each of these phloem parenchyma, they are connected to one another through plasmodesmatal connections. So one cell is connected to the other through connections called as the plasmodesmatal connections. So, and the phloem parenchyma, it usually helps in the storage of food materials. So they remember I told you parenchyma means the main function of parenchyma is to store food. Just like that, phloem parenchyma also helps in the storage of food materials such as resins, latex and mucilaginous substances. So all the resins, that is the gummy substance, the latex, as well as the rubbery substance and the mucilaginous substances, all those are stored in the phloem parenchyma. Why so? Because phloem parenchyma is one of the tissues which help in the conduction of the food, therefore. And phloem parenchyma, it is absent in most of the monocotyledons. So this is one of the characteristic feature of the species of plants coming under the monocotyledons, wherein most of the species under the monocotyledons, they do not have phloem parenchyma. And also gymnosperms have all these, but they don't have the sieve tubes and the companion cells. Instead, they have the sieve cells and they have the albuminous cells. So these are some of the characteristic features of the phloem tissue. So next, moving on to the phloem fibers. So phloem fibers are also called as the bast fibers. Just like the xylem fibers, these are also thin, narrow, elongated, and they have tapering ends, and they are made up of sclerenchymatous cells. So therefore they are dead. The phloem fibers are dead tissues. Why? Because they are made up of sclerenchyma. So they are made up of sclerenchymatous cells. Made up of sclerenchyma, that is why. And they are very long, narrow, elongated, and they have tapering ends. So generally they are absent in the primary phloem, but they are usually found in the secondary phloem. So primary phloems, these sclerenchymas are usually absent, but when it comes to secondary phloem, these phloem fibers are usually present, and they are much elongated. As I told you, they are unbranched and they have pointed or needle-like apices. That is, they have pointed ends. So two ends, if you see, they are either pointed or they are almost needle-like. They are pointed or they are needle-like. So both the ends, even the upper end also, it is one and the same. They have a tapering, pointed or needle-like apices. So apices is nothing but ends or tips. The tips are pointed, and the cell wall is usually quite thick. So cell wall of phloem fibers is very, very thick, and at maturity, these fibers, phloem fibers, usually lack the protoplasm, or the protoplasm is lost when the phloem fibers reach them maturity, and therefore they become dead. Remember, in sclerenchyma, they do not have the protoplasm. Therefore, the cells of the sclerenchyma are dead. So similarly here, at the beginning, the phloem fibers in the primary tissues, they usually living. Why? Because they will have the protoplasm. But eventually, as the phloem fibers mature, what happens? These phloem fibers, they will lose the protoplasm that is present in them, and therefore they become a dead tissue. And these phloem fibers are nothing but found in the fibers of jute. That is why they are very, very rigid, they are very rough. They are present in the fibers of jute, in the flax and hemp, and therefore they are used commercially in manufacturing ropes or making bags, certain other accessories and all that. They are being mats and for all that. So they are usually gunny bags. So various products are made, commercially important products are made out of these phloem fibers. So almost all the plants have the phloem fibers in them. So next, moving on to the primary phloem, like how in the primary xylem had the protoxylem and the metaxylem. Just like that, the primary phloem has protophlorium and metaphlorium. So protophlorium means they are the first formed primary phloem. After the formation of the protophlorium, then forms the metaphlloem. So they are the first formed primary phloem and they consist of narrow sieve tubes. So they are usually made up of narrow sieve tubes, and metaphlloem, they are the later formed phloem and they have bigger sieve tubes. So that is why they appear bigger in size when compared to the protophlloem. So you can see the protophlloem, they are usually smaller, and the metaphlloem, they are usually bigger. So this is about the protophlloem and the metaphlloem. So I hope you understood today's session well, wherein we discussed about the permanent tissue. So in the previous sessions, before the session, we had talked about the meristematic tissue, and again in meristematic tissue, they are divided into the primary tissues and the secondary tissues. So in the secondary tissues, they were, and also we talked about the permanent tissues. So permanent tissues are the completely matured form of cells. So in these permanent tissues, again, there are the simple tissues and the complex tissues. So simple tissues, parenchyma, the collenchyma and the sclerenchyma, and the complex permanent tissues are the xylem and the phloem. So again, in the xylem, we studied about the primary xylem, and in the phloem, we studied about the primary phloem, and also we studied about the components of or the elements of the xylem, and also the components or the elements of the phloem. So I hope you understood today's session well. In the next coming sessions, we shall study about the other tissues, that is the tissues that are present, how to differentiate between the cells that are present in a dicot, a monocot root, a monocot stem, and so on. So we shall meet in the next coming session. Thank you.